Welcome to Neet. I am Sheikh Altaf. Today we will discuss molecular ways of inheritance. The two scientists they prove that replication of DNA is semi-conservative. Semi-conservative means after replication, the two daughter cells have two templates. Out of the two templates, one is parental template and another is new. So that means every replication they produce that one old and one new. That's why they call it semi-conservative. See that they conducted one experiment. The name of scientist Meselson and Stahl in 1958. They took one bacteria, E. coli bacteria, and E. coli bacteria they grown in the medium contain ammonium chloride. In ammonium chloride, he took that N15. What is N15? It is a heavy isotope of nitrogen. They took the ammonium chloride, the nitrogen contain heavy isotope, and the E. coli bacteria culture under this heavy isotope of nitrogen. After culture, he find out that the DNA is heavy isotope and N15 it is not a radioisotope. So you should not be confused about the radioisotope and heavy isotope. Heavy isotope it is different from the radioisotope. Then how can you measure the density of nitrogen? For that he took the cesium chloride. Cesium chloride is the chemical they add into the test tube, and after that he added N15 bacteria, that is heavy isotope nitrogen of bacteria. After he just make into centrifugal force. After centrifugal force, he observed that the heavy nitrogen will go to the bottom of the test tube. So this line indicates for that heavy isotope of nitrogen. So like that he can find out that here in his experiment. So heavy isotope, the cesium chloride will be identified that heavy isotope DNA is bottom and hybrids are 15-14 is middle and lights are 14-14. So according to that centrifugal force, they can separate the DNA into three stages: heavy, hybrid, and light. So first, what he done in first generation, he took the N15 nitrogen into the bacteria contain the light nitrogen. Light nitrogen means we can take the E. coli bacteria. E. coli has the DNA. The DNA has 14. Nitrogen is light. After this culture, after 20 minutes, you know the bacteria can complete the recombination in 18 minutes. 18 minutes is the recombination completed. But you need the 2 minutes for further replication or further reproduction. So after 20 minutes, what will happen? The heavy nitrogen template of DNA is combined with light N14 template and they produce one hybrid. That means we can observe that black color is the heavy nitrogen and green color is the light. So from the coloration you can understood that first generation after 20 minutes he find the two bacteria and the two bacteria have one is heavy, another is light. So heavy and light is there means it will be hybrid, and in the test tube you add the same cesium chloride and under centrifugal process he observed the line is came middle. You can see the middle line is there. Here N15 is there and N14 is there. So both templates are one is N15 heavy, another is light. So hybrid. So like that he can find how many number? Two pieces of DNA. Both are hybrid. Hybrid is nothing but one is old and another is new. This is new, this is old. So mixing after they form the hybrid. Same experiment. Again continue. Next 20 minutes. Next 20 minutes, 20 plus 20, 40 minutes. After 40 minutes, how the culture means same thing. He took this hybrid DNA. Hybrid DNA he took under the culture. Under culture of E. coli, the E. coli bacteria has light DNA is there. This is 14 and 14N. So this 14N, this part, this hybrid DNA, he take inside the E. coli bacteria, and E. coli bacteria cultured with this hybrid DNA. After that, what happens? It can produce that here N15 plus N14, one hybrid, and there is N14, one light. Like that, this bacteria produce again two types of DNA in second generation after 40 minutes. In second generation, one bacteria he produce one hybrid, another is light. Likewise, again you take this, you took this hybrid DNA and culture into the same another E. coli bacteria. Another E. coli bacteria, it has two light nitrogen DNA is there. After same thing will be again 15, 14, 15, 14 hybrid one. Again next to this 14, 14 another E. coli bacteria to produce light. So this is another bacteria. They produce the two DNA. Out of the two, one is light, another is a hybrid. This is again same thing. The two DNA they produce after multiplication of this E. coli bacteria. So out of this second generation after 20 minutes, I mean 20, 40 minutes, they produce the 4 DNA or 4 E. coli bacteria. Out of these 4 E. coli bacteria, you can see that here, the 2 are hybrid and 2 are light. After keep in the test tube and centrifugal process, he observed the 2 lines are there. The middle is there 15 and 14 dark, and this hybrid, and mouth of this test tube they form 14, 14. That means in after 40 minutes they observe that 2 hybrid and 2 light. The meaning of hybrid is nothing but the DNA replication is semi-conservative, 50-50% is there. Like that you continue that. Suppose you will continue the same thing. See that here after 40 minutes, after again you add the 20 minutes. After adding the 20 minutes, 20 plus 40, 60 minutes. So 60 minutes means 1 hour. So after 1 hour, we can observe how many DNAs. Here how many are? 1, 2, 3, 4 is there. And the 4 DNA cultured in the 4 light bacteria. 4 means that the second generation is 4 into 2. So 8. So you can observe that here how many are? 8. So 8 out of 8 DNA. Out of 8 DNA. Again will be, they produce the 2 hybrid. Out of 8, they produce the 2 hybrid. That means 8 minus 2. So 8 minus 2 is equal to 6. So 2 hybrids you observe after 60 minutes or 1 hour, and remaining 6 are light. 6 are light. 1, 2, 3, 4, 5, 6. Now this 1, 2, 3, 4, 5, 6. 6 you observe that light, and he find out that both will find out that 2 hybrid. So 66 is to 2 is equal to 3 is to 1. That means the 3 is to 1 ratio he observed in that after 60 minutes. So 60 minutes the hybrid will be 2 and light 6. So suppose you continue same thing. In the textbook they given NCERT book after 80 minutes. So after 80 minutes, how many light and how many hybrid DNA produce? So after 80 minutes means 60 minutes they form the 8. Again that multiplication, each bacteria undergo replication or reproduction to produce 2 bacteria. So here 8 is there, means after 20 minutes it will come to 16. So here you can find that number of DNA is 16 after 80 minutes. You know the every generation they produce two hybrids. So every generation they form the two hybrids. That's why after 80 minutes Out of 16 DNA, 16 minus 2. 16 minus 2 means 14. So 14 DNAs are light and 2 DNA hybrid. So you can calculate that suppose the ratio they will ask light is to hybrid. That means light is to hybrid. So what is the ratio of light is to hybrid after 80 minutes means you can calculate the light or 14 is to hybrid 2. So 14 is to 2 is equal to, you can just divide by 2, then it comes to 7 is to 2. So 7 is to 1. So now you can say that the ratio of light and heavy DNA after 80 minutes is 7 is to 1. So numerical question they will ask. So what type of numerical question ask means they will ask in 1 hour. According to that Meselson and Stahl experiment, the DNA culture under that light and heavy DNA after 1 hour. 1 hour means 60 minutes. After 60 minutes or 1 hour, how many hybrid means you can find out that 8 minus 2, 6. 6 are light and 2 hybrid. So any numerical question they will ask means you can find the first generation 2 are hybrid, second generation 4, 2 are hybrid, 2 light, third generation again 8, out of it 2 hybrid, 6 light, and next generation 18, 18 means 18 minus 2, 2 hybrid and light. Like that, how many generation continuation? Every generation he observed that 2 hybrids are. That means he finally proved that the DNA replication is semi-conservative.